Dear students, you are welcome in this class. Now we have very interesting topic and you may be interested to know more about this topic, that is movement of nutrients in soil and their uptake by plant roots. So this is really very important topic and somewhat difficult to understand also. So you need to be a bit careful while I go through this subject. And movement of nutrients here means how the nutrients are reaching the plant roots from the soil solution. As you may be knowing that soil solution is the have the nutrients in available form, and those nutrients will go to the plant root. And sometimes plant root can also come in contact with the soil solution and directly absorb the nutrients. And sometimes plant roots can also reach the exchangeable site in the soil and it can directly contact the nutrients. But mostly nutrients move from soil solution to the plant root, and then plant roots take up nutrients inside them. And this mechanism is very difficult to understand because number of theories are there in this nutrient uptake and nutrient transport. So learning objective is to learn the mechanism of movement of ions to plant roots, how ions or nutrients are reaching the plant roots, describe the process of nutrient uptake by the plant roots. So these are the two clear-cut objectives, and there may be some active transport of nutrients and there may be passive transport of nutrients. Active transport of nutrient or ions, you can also call it ions, movement of molecules or ions across a membrane from a region of lower concentration, to a region of higher concentration against the concentration gradient, and then you require the cellular energy. So with the help of energy, the nutrients can move from lower concentration towards higher concentration. And you have apoplastic movement and symplastic movement. Apoplastic means apoplast is the space outside the plasma membrane. So at the outside in the plant cell, you have cell wall, then cell membrane, and then various organelles in the cell membrane, surrounded by cell membrane, these organelles. So you can see there are some interspaces between cell wall and plasma lemma or cell membrane. So apoplast is the space outside the plasma membrane within which materials can diffuse freely from one cell to another cell. It is slowed down by the Casparian strip in root. Nutrient carriers, a theory explaining the transport of nutrients from the surface of roots into the root cell, through various nutrient carriers occurring by means of carriers. Plasma membrane, microscopic membrane made up of lipids and proteins forming external boundary of the cytoplasm of a cell or encloses a vacuole. That is your plasma membrane or cell membrane, and also called as plasma lemma. So dear students, you can see the structure of plasma membrane. This is very important. You can get any plant physiology book and just study the structure of plasma membrane. This is really important for agronomy student also. And then plasmodesma or plasmodesmata, microscopic channels on the cell walls of plant cells, helping the movement and needful communication between the plant cells. So plasmodesma or plasmodesmata, these are the small holes in the cell walls through which materials pass from one cell to another cell. Symplastic movement, the diffusion of water or low molecular weight solutes in the inner side of the plasma membrane is called as symplastic movement. Nutrient supplying power of soil, the capacity of the soil to supply nutrients to growing plants from the labile, exchangeable, and moderately available form. Diffusion coefficient. This is proportionality constant that indicates the ability of a material to allow gases and ions to flow under a partial pressure or concentration gradient. This is diffusion coefficient. Nutrient uptake is the process of ingesting. Ingesting means taking inside, process of ingesting nutrients by plant roots, or it is the transfer of the nutrient ions across the soil root interfaces into the plant cell. Now, labile, a substance that is readily transformed or used by microbes, is labile. Your nutrients have several forms: labile form, non-labile form, like this. Nutrient concentration and nutrient content. So concentration expressed in gram per kg or milligram per kg or of dry or fresh weight. So you can get this concentration weight by weight. When you have weight by weight or volume by volume, then you call it concentration. And when it is weight by unit area, then content. So it is better to write nitrogen concentration in plant than nitrogen content in plant. So this is the difference in concentration and content. For nutrients, we should always write concentration, not the word content. Content is for if you have kg per hectare, means weight per unit area, then it is content. And weight by weight, you are reporting anything, then it is concentration. Nutrient efficient plant. A plant that absorbs, translocates, or utilizes more of a specific nutrient than another plant under conditions of relatively low nutrient availability in the soil. Then that is nutrient efficient plant. So nutrient efficient plant can be for one nutrient. It cannot be for all the nutrients that you will have one plant which is efficient. For example, certain varieties of wheat or certain varieties of rice can better absorb zinc, can better absorb zinc under zinc deficient conditions. Then that will be zinc efficient plant. Means under stress condition, if a plant is able to take up the nutrient or extract the nutrient and without showing any deficiency, then that is your nutrient or zinc efficient plant. Now, movement of nutrients from soil to roots. How nutrients will reach the plant roots, how they will come close to the plant roots for their uptake. So nutrients move by three different mechanisms in the soil. One is root interception, where roots are reaching to the nutrients. Mass flow, where nutrients are moving in mass along with water. Then diffusion, movement of nutrients from higher concentration to lower concentration. So in this picture, you can see that mass flow, they are in mass, they are in large numbers, these ions or molecules, and they move along with water. And you can see this is mass flow, and this is happening in soil solution. So how they are going to the plant roots in soil solution. Then interception also, these roots can directly come in the contact of the nutrients. And then diffusion. Diffusion is again from higher concentration, movement of nutrients from higher concentration to lower concentration. So that way they are coming to the plant roots, and then they can enter the plant roots and they can go up for transport. Movement of ions and nutrients to the roots by interception, mass flow, and diffusion. So we'll see one by one. Now, root interception. Exchange of ions through physical contact between root and mineral surface. So root can directly come in the contact of minerals, enhanced by increased root biomass and proliferation. So if you have more root biomass or more proliferation or expansion of roots, then they will come in more, they will come in larger, they will come in contact with the larger volume of soil. So you can expect more root interception if your root growth is more. Roots extend to soil solution, soil surfaces to access nutrients. So wherever nutrients are there in soil, if roots go there, if roots are able to reach there, then the nutrients can be taken up by roots by interception. Hydrogen ion adsorbed on surface of root hairs exchange with ions held on the clay and organic matter surfaces. So sometimes these hydrogen ions comes out of the cells, and they are attached to the surface of the root, and then they can be exchanged with ions held on the clay and organic matter surfaces. This is by root interception. Ions held by electrostatic forces at these sites oscillate with a certain volume. Means when interception happens, some nutrients may be around the root, they may be very near to the root, and then some oscillation. They will oscillate, they will move, go forward, come back, go forward, come back. It is oscillation. So when the oscillation volume of two ions, hydrogen ion and potassium ion, overlap, when they exactly come close to each other, when they touch each other, then ion exchange occurs. When they touch each other or overlap each other during oscillation, then root interception can happen, or ion uptake can happen. Calcium ion on clay surface could be absorbed by roots. So if it is on the exchangeable sites, this calcium or potassium, even potassium is by oscillation, and calcium can be directly taken up by roots present on the clay complex or present on the colloid. Roots usually occupy 1 to 3% of the soil volume. That is in 0 to 15 centimeters. Now see how this oscillation happens, how the exchange of potassium can happen with hydrogen. You know, hydrogen can be released or can be uh, exuded by the roots, and potassium are present in the on the clay complex or in the soil solution. They can be in the on the clay complex or in the soil solution. Conceptual model for root interception or contact exchange of nutrients between ions on soil and root exchange sites. So you can see this potassium ion is on soil and hydrogen ion is on root. Overlapping oscillation. This is oscillating, and there is some overlapping. Cause exchange of hydrogen ion on the root with potassium on the clay mineral. So they will exchange. So this is how root interceptions take up the ions directly from the clays. Now see, this is very interesting fact that which nutrient is moving or reaching the plant roots by which method. Significance of root interception, mass flow, and diffusion in ion transport to corn plant. How ions are transported in the soil. So first you read the note. The contribution of diffusion was estimated by the difference between total nutrient needs, means how much total nutrients were required by the crop, and the amount supplied by the interception. Out of the total nutrient applied, recommended nutrient apply, how much was moved by interception and mass flow, and also diffusion. If root interception plus mass flow is more than or equal to 100%, then diffusion is zero. So they have fixed the diffusion as zero when total of interception and mass flow is more than or equal to 100%. This is just relative uptake by interception, mass flow, and diffusion. Take the first case. Take the first case of nitrogen. Nutrient required for 200 bushels per acre of corn. This is the quantity of the corn, targeted yield of the corn. Say, it was 200. And root interception, mass flow, and diffusion. So here you can say nitrogen is totally removed by mass flow. Say 99% is by mass flow, and root interception is 1. And similarly, you can see phosphorus, it is mostly by diffusion, 94. And see potassium, it is by diffusion again. And see calcium and magnesium, diffusion is 0, and it is largely removed by mass flow and also by root interception. Your sulfur is by mass flow. Your copper is by mass flow. Your zinc is by three different methods. One is 45 diffusion and then mass flow and also by root interception. Boron is by mass flow. So at least for nitrogen, phosphorus, potassium, and calcium, magnesium, you can remember that which mechanism is more important for movement of nutrients from soil solution to the plant root.
Sir,
Yes?
yes, sir. In case of calcium, the percentage is very high. Uh, So it should be under 100 in case
No,
of.
actually, you would surprise, you will think that how it is more than 100%. So this is in relation to diffusion. This is in relation to diffusion. That is why I started from here. If root interception plus mass flow if they make together more than 100%, then diffusion is zero. So they calculated it keeping the diffusion zero.
Okay, sir.
So these are relative. Now you see root interception continues. A root interception may be enhanced by mycorrhiza. So mycorrhiza are very, very important in nutrient mobilization as well as nutrient uptake. More benefits to plant in, particularly more benefits in infertile soils where your nutrients are low, nutrient content is low. Here you need to remember that mycorrhiza help in the uptake of phosphorus and many other nutrients. It is not just phosphorus, but mycorrhiza also help in the uptake of zinc, uptake of sulfur, almost all the nutrients. They are important for their uptake. So mycorrhizal colonization enhanced in slightly acidic soil pH. So they are more active when soil pH is acidic and low P, low phosphorus is there, adequate nitrogen and low soil temperature. So these are the ideal conditions for the growth of mycorrhiza. Slightly acidic pH, low phosphorus, adequate nitrogen, and low soil temperature. The hyphal threads act as an extension of plant root system, greater soil contact. So they are actually kind of mutual relationship where this fungus is getting some you say carbohydrates. I think it will get carbohydrates or energy sources, and in turn it will give some nutrients and water to the plant. It will give nutrients and water to the plant, and this exchange of nutrients happens in which part of the mycorrhiza. See, they will exchange nutrients and water will be provided by mycorrhiza to the host, to the plant, and host will give it carbohydrates for its development. So what are the sites of mycorrhiza responsible for exchange of materials
So
between
arbus,
host? Huh?
so arbuscle.
Exactly. So these are the arbuscules where this exchange happens, and these arbuscules are inside the cell. They are inside the cell or intracellular. Within the cell, they enter the plant cells, root cells, you can say, and where the exchange happens. So you can see there are two groups of mycorrhizas, ectomycorrhiza and endomycorrhiza. They are ecto and endomycorrhiza. Ectomycorrhizas are mostly in tree species, on the trees, temperate zone, mostly in temperate zone. Also in semi-arid zone, endomycorrhizas are more widespread. They are mostly our most of our field crops. They have endomycorrhizas, and temperate trees have ectomycorrhizas. And besides nutrient uptake, they help in the nutrient uptake. Besides this, mycorrhiza also help the plants in tolerating other kind of stresses also. Roots of most agronomic crops have arbuscular mycorrhiza or AM fungi. So these are actually endomycorrhizas. These AM fungi are endomycorrhizas. It grows into the cortex. They enter the plant cells. They enter the root cells. Inside the plant cells, a small structure arbuscules are the transfer of nutrients from fungi to host plant. So you can see here, photosynthetic product or carbohydrates are given by plant to the mycorrhiza, and water and mineral is given to the plant by the mycorrhiza. AM fungi, arbuscular mycorrhiza fungi, large nutrient absorbing surface by fungi can be up to 10 times that of uninfected roots. So they increase the absorptive area of the roots. Fungal hyphae can extend up to 8 centimeters into the soil, thus increasing the absorption of nutrients. And its size is very smaller, very fine, even than the root hair. So they can go to the spaces or micropores in the soil where root hair cannot enter. So they increase the absorptive area and they interact with more soil volume. Enhanced the uptake is the primary cause of improved plant growth. So basically, they improve the phosphorus uptake, but that does not mean that they help in the uptake of phosphorus only. They also increase zinc, sulfur, and other nutrients uptake also. Now you can see very interesting data. Effect of inoculation of endomycorrhiza and phosphorus on nutrient content in corn shoots. So see, this is a maize crop and content of phosphorus in microgram in the shoots. So you can see no phosphorus. When phosphorus was not added, then what happened to different nutrients? So here 750 microgram was phosphorus without mycorrhiza. But if you have added mycorrhiza, then it has increased, almost doubled, when there was no phosphorus added to the soil. See potassium. Potassium uptake under control was 6,000, but it has increased if you have mycorrhiza. So here you can see that phosphorus, potassium, calcium, magnesium, zinc, copper, manganese, and iron uptake increased if you have mycorrhiza. So this is number one conclusion, that if you have mycorrhiza, even without adding phosphorus to the soil, it increases phosphorus uptake, potassium uptake, calcium uptake, magnesium uptake, zinc, copper, manganese, and iron uptake. You can see this here. What happens when you add phosphorus to the soil as a fertilizer? It was added. And again, two kind of condition, no mycorrhiza and mycorrhiza. So you can see even under conditions of phosphorus addition, you have increased uptake of all the nutrients. Increased uptake of all the nutrients. So this table clearly establishes the fact that mycorrhiza help in improving the uptake of not just phosphorus, but potassium, calcium, magnesium, zinc, copper, manganese, iron, and many other nutrients under no phosphorus condition or even under conditions where you add phosphorus to the soil. So in any case, you can see the mycorrhiza are effective. Now we go after root interception, we go to the mass flow. Ions of soil solution are transported to root as a result of water uptake, transpiration by plant, evaporation, soil surface, and percolation of water in soil profile. You can see water moves in the soil, and along with that water, nutrients also move in larger quantities, which is your mass flow. Important factor in supplying nutrients to plants. The quantity of nutrients reaching roots is determined by rate of water flow or water consumption and average nutrient concentration in the soil water. So mass flow is important. You see under dry land conditions. Under dry land conditions, the quantity of nutrients applied or required is much less than the irrigated conditions. So therefore, under dry land, you cannot add more quantity of nutrients or higher nutrients because water shortage is there and nutrients will not move in mass to the plant roots. So that is also reason. And you must be knowing that nutrient water interactions are significant. Very high interaction happens between nutrients and water in the soil. Now, a mass flow can supply an excess of calcium, magnesium, and some micronutrients and more soluble nutrients like nitrate, chloride, and sulfate. So these are the ions which move through mass flow. As soil moisture is reduced, water transport to the root surface decreases. Also reduce at low temperature. If you reduce the temperature, the mass flow will also reduce, mainly because of reduced transpiration and evaporation. Now, third one is diffusion. Diffusion through which the nutrient from soil solution will go to the plant roots. So ions move from an area of high concentration to one low concentration. Roots absorb nutrients from surrounding soil solution. The nutrient concentration at root surface decreases compared with the bulk soil solution concentration. You know, when the nutrients are reaching the plant roots, they are taken up by the roots and the low concentration happens there. But the soil which is away from the roots, there you will get higher concentration of the nutrients. So movement is from higher concentration to lower concentration. Nutrient concentration gradient is established that causes ions to diffuse towards the root. Most of the phosphorus and potassium move to the root by diffusion. And sometimes back diffusion can also happen. That is not very regular, but you will find back diffusion means there is very good supply of nutrients to the plant roots by mass flow or by diffusion or by both. Then concentration of nutrients may be for a shorter period higher near the root compared to the bulk soil. So it can go back. It can go back to the bulk soil. So that can also happen, back diffusion. So diffusion occurs based on the fixed law, which is given by the equation F is equal to minus D dC by dx, whereas F is the diffusion rate, quantity diffused per unit cross section per unit time. This is the flow. dC by dx is concentration gradient and C is concentration, D is diffusion gradient and X is the distance. So this is the fixed law and this is used to measure the diffusion rate of the diffusion. Ion uptake by roots create or maintain diffusion gradient is strongly influenced by temperature. So diffusion is also affected by temperature, strongly by temperature, within the range of 10 to 30 degrees centigrade. An increase of 10 degrees centigrade usually causes the rate of ion absorption to go up by the factor of 2 or more, or it is double. Means when you increase the temperature from 10 to 20, the diffusion rate is doubled, or from 20 to 30, diffusion rate is doubled, or more. So, but it is up to 30 degrees centigrade. Diffusion slower under most soil conditions and occur over very short distances. This is the limitation, that it is slow under most soil conditions and occurs over very short distances. You cannot take ions to 10 centimeter or 5 centimeters. The distances are small. Typical diffusion distance are 1 centimeter for nitrogen. You can remember these values sometimes in quiz. In quiz, these kind of questions are asked. 1 centimeter movement of nitrogen by diffusion, 0.2 centimeter for potassium, and 0.02 centimeter for phosphorus. Phosphorus is the most lazy nutrient. It moves very slow and in very less quantities. Phosphorus. Roots do not absorb all nutrients at same rate, causing certain ions to accumulate at root surface. Certain can accumulate at root surface. So therefore, some back diffusion can happen. It results in back diffusion where the concentration gradient is away from the root surface and back towards the uh, bulk soil solution. Nutrient diffusion away from root is much less common than diffusion towards the root. So that is true, but sometimes you can find back diffusion also. Importance of diffusion or mass flow in supplying ions to root surface depends on ability of solid phase of soil to replenish or buffer the soil solution. This is also important because your nutrients by diffusion or even by mass flow are coming from the basic source is your soil solution. From soil solution, they are going to the plant roots. If there is shortage of nutrients in the soil solution, how come it can supply the nutrients to the plant root? So therefore, it should be the soil solution should be effectively replenished by the nutrients from other sources. What are other sources? Your exchangeable sites or exchangeable cations or ions, or it may be directly from the solubilization of the minerals. Minerals can decompose and they can supply nutrients to the soil solution. So ion concentration influenced by the types of clay minerals in the soil and distribution of cation anions on the cation exchange capacity or anion exchange capacity. This is also important, how these cations are released from the cation exchange, from the exchange site. Exchange site. For example, the ease of replacement of calcium from colloids varies from this order. Peat. If you have calcium on peat, it will be easily released. Then kaolinite, and then illite, and montmorillonite. So if your soil is dominating this montmorillonite clays, then your calcium will be absorbed tightly, more tightly, and release will be less. But if it is peat or kaolinite, here your calcium will be loosely absorbed, and it can be easily replaced, and easily it can diffuse to the soil solution. And 80% calcium saturated 2:1 clay provides the same percentage of calcium release as 35% calcium saturated kaolinite or 25% calcium saturated peat. So this clearly shows that peat can easily release calcium. And then next is your kaolinite, bit difficult, not as good as peat, and montmorillonite is in the end. It absorbs calcium more tightly.
Sir,
Yes,
so but we know that the cation exchange capacity of montmorillonite is much higher than kaolinite. Then it should replace more calcium.
see, this is the important point. Your point is that overall, the montmorillonite will have more cations, not just calcium. It will also have other cations. Similarly, peat, kaolinite, and illite will also have other cations. So I think this is the characteristic. No doubt, montmorillonite will have comparatively more cations, but they are not easily replaced. They are not easily lost. They are not easily removed from the exchangeable sites as compared to peat or kaolinite. So this is actually order of their release from the exchangeable site or replacement. Uh, uh, Montmorillonite is having them this calcium very tightly, so less are released.
Okay,
Less
sir,
are
clear.
released. Now, ion absorption by plants. So up to now, you have seen that anyhow, whether root is reaching the ions or molecules, or the solution or nutrients themselves are going to the root. Nutrients are coming near to the roots from soil solution. They are reaching by interception or by mass flow or by diffusion. Now they have come in the contact of the roots. So which is not the complete uptake. Uptake of nutrients will happen when these nutrients enter into the inside the cell. When they are inside the cell, then we can say nutrient uptake is there. So once nutrients are at the root surface, they enter the outermost root cell. So they will make beginning from the epidermis, the outer layer. From there, they will enter the cell wall, and then other cortex, and so many things are there. You will learn now. So once nutrients are at root surface, they enter the outermost root cells where they are transported through the plant. And inside there will be transport, horizontal transport as well as vertical transport. So first horizontal transport is important. Through horizontal transport, the nutrients or ions will reach the xylem. They will reach the xylem, whether it is water or nutrients. Both have to pass through cross section, and then they will reach the xylem. And as you know, the food material moves in phloem. So ion transport out of the root also occurs and can influence rhizospheric pH. So sometimes the ions may get out of the plant roots also. Some ions like hydrogen ions, they may come out. There may be some other ions also which may come out from the roots, and they can influence the pH of the soil. Thus, it is important to understand how ions and water move into the plant roots. And movement of ions into the plant roots is your uptake of nutrients. Now, transport system. Before we go into the actual nutrient uptake, let us understand transport systems in plant, how these nutrient and water and food materials are transported from one part to another part. So plant stems and roots transport substances around plant. Two types of transport systems are there. I think each one of us knows about xylem and phloem. The xylem also have variety of cells. Different kind of cells are there in xylem. Similarly, phloem also have different kind of cells. Xylem transport water and minerals as clearly known to you. Phloem transport sugars and amino acids dissolved in water. In soluble form, it is transported by phloem. Or you can say synthesized food materials are transported by the phloem. And raw materials like minerals and water are transported by xylem. These are distributed differently in roots and stems. They are distributed differently in roots and stems. Differently means in some parts they will give more, in another part they will give less. You know, for example, in seeds, sometimes you will get more proteins than the leaves. So this is given by nature or controlled by genes. In the root, the xylem forms a central column. It forms a solid support. You know, or phloem. They are through the plant system, starting from tip of the root to the tip of the leaf or tip of the stem, or these two vascular bundles are basically in continuum. They are in continuum. They are continuously. They are sometimes their shape and size and their cells may change, but they are in continuity, so that the materials are distributed in plants uniformly, not uniformly to all the parts. If the stem, the transport tissues of the xylem and phloem are grouped into vascular bundles, they are called as vascular bundles. The phloem is towards the center, outside is the xylem. You know about these things since your high school, I think. So you can see the transport system in plants, very rough, or pictures are there to have more clarity. So you can see central part you have xylem, and then this is surrounded by phloem. This yellow one is your phloem, or you can see more clearly here the vascular bundles, and one yellow one is your phloem outside, and inside this is your xylem. So xylem transport water and mineral nutrients from roots of the plant stem and into the leaves, which is known as kind of vertical transport. In a mature flowering plant, most of the cells that make up xylem are specialized cells called vessels. Xylem vessels are responsible for transport of nutrients or water. Vessels lost their end walls, so the xylem forms a continuous hollow tube. They make a pipe. They make a pipe of vessels. Become standard. These vessels become standard. They become more strong by a chemical called lignin. So cells are no longer alive. So lignin is accumulated inside. Lignin gives strength and support to the plant. Well, we call lignified cells wood. Lignified cells wood. You can see more about xylem. It is like continuous tube. So transport in the xylem is a physical process. It does not require energy. This is simple physical process. You know, transpiration pull kind of things are there. And to have more clarity, you can see some more pictures I wish to show you. So this is solution, or you can say these are the molecules or nutrients. They are taken up by root here. They pass through the different cell layers in the root, and finally they reach the xylem vessel. This is horizontal movement. This is horizontal movement. So soil water, higher water potential is there, and root here, lower water potential. Now you see more clearly how the nutrients move from plant roots up to the leaf. So you can see these are soil particles. Here you have nutrients, water surrounding soil particle, water is there, and then you got the root or root hair part, and nutrients are taken up, and they are passed on to the stem from the root. In the root, they should reach the xylem, and once they reach the xylem, cross section or horizontally, they can go to xylem horizontally, and then they start their vertical journey. You can see in root they are horizontal. They are entering the xylem, and once they have entered the xylem, they go upward. They go upward. Xylem contains a continuous column of water. So here your nutrient will go, and then your branches may come. Your leaves may come on the stem. Then it can have lateral movement or side movement because it is in continuity. Xylem is in continuity. Then again you will have branches and then leaves. So it will go to the leaves, up to leaves, this xylem. So leaves also have your xylem. So you can see it started with horizontal and then go and it reaches up to the leaf. So this is here the nutrients will be accumulated. Nutrients or water will be accumulated and then again evaporation and so on. So this is very simplified diagram to show you transport system in plants, transport of nutrients. To have more clarity, you can see different zones of roots. So you can start from the root tip. This region is your root tip. Apical meristem is the tissue through which the root grows. And then epidermis is the outer layer of the root, epidermis. Then inside it, there is one endodermis, one layer of cells. Then pericycle, then phloem, then xylem. Xylem are the innermost, and you can see cortex. Cortex tissues are there. These are cortex. Root cap is zone of elongation. This one is zone of elongation, which is above the apical meristem. The zone of maturation is there, and here you got root hairs. So what happens? Many times, students think that nutrients are taken up only by the root hair. That is true, but besides root hair, nutrients are also taken up or absorbed by other portion of the roots also. It is not just root hair which are responsible for nutrient uptake. Even epidermis can directly absorb the nutrients. So what are root hairs? They are extension of the epidermis. They are extension of the epidermis. So you can see some more pictures to have more clarity. You can see root hairs coming from epidermis, then endodermis is there. These are your cortex tissues, cortex cells, and then pericycle is there, then primary xylem, and surrounded by phloem, primary phloem. You got one cambium, vascular cambium is there between two xylem and phloem. You get vascular cambium, etc. You can see different parts of cross section of the root. Now see very important data, different areas of roots for nutrient absorption. For example, in barley, calcium is absorbed mainly by root apical meristem apical region, iron root apical region. In corn, iron is absorbed by entire root surface equally. Several crops, potassium, nitrate, ammonium, and phosphate, entire root surface, root elongation zone. So overall, the purpose of this slide is that nutrients are absorbed by all the parts of the root, depending upon the crop or kind of plant and the kind of nutrient. So they are absorbed by whole root. Of course, the uptake is more effective and more speed from the through the root here, but that does not mean that it is the only way to remove the nutrients. Now you see nutrient uptake by plant roots. We come to the main topic now. Nutrients come in contact with the plant root by root interception, mass flow, and diffusion. This is already clear to you. These nutrients will now enter the plant roots and get transported into the plant cell. Now they must enter here, and they have to reach up to the xylem. How they can reach? There are number of theories, number of ways by which plants can enter the root cells, and then they can reach up to the xylem. So these nutrients will now enter the plant cells and get transported into the plant cells. Nutrient uptake by plants can be addressed in two parts. One is uptake of nutrients ions by roots from the rhizosphere. First thing is ingesting them, taking the nutrient inside. And then when they have been taken inside the root, then how they travel to the xylem. These two things are important. Uptake of nutrients ion by roots from the rhizosphere. Second is uptake and transport. So transport will be horizontal transport. Basically, it will be horizontal transport up to the xylem in the plant cells. So nutrient and water uptake happens at the root hairs and the rest of the root epidermis. The whole root absorbs the nutrient as well as water. It is not just root hair. Now, uptake from soil solution can be described by passive and active processes. So uptake from rhizosphere, or you can say from soil solution, or when the nutrients have reached by mass flow or diffusion to the plant roots, then it can be taken up by plant through passive and active processes. Ions passively move to a boundary through which ions are actively transported to organs in plant cells. So we will see all these theories. Solution composition. Ion concentration outside and inside of the boundary, controlled by different processes, each essential to plant nutrition and growth. Now, there are three important, before you try to understand the exact mechanism of the nutrient uptake, we should see the three things are important when they enter the cell, entry into the cell. One is adsorption. Adsorption means the nutrients should come to the surface of the root. That is also part of the nutrient uptake. When they come to the surface of the root, and then from surface it goes absorption. They go inside, and then inside the cells they get accumulated. So these three things are important in nutrient uptake: adsorption, absorption, and accumulation. Friends, I tell you, this subject is very, nutrient uptake is very complex. It is not very easy to understand. It has got a lot of confusion, and so many things are involved that it is really difficult to explain exactly the nutrient uptake process. So therefore, I'm taking the things in pieces so that in the end we can join all these pieces and try to understand. So more you read about it, more you get confused, this nutrient uptake. Anyway, now you see adsorption, absorption, and accumulation. The uptake of cations begins with the adsorption of the cations to the cell wall, where polysaccharides carry negative charges. So if you know the structure of the cell wall, it has got polysaccharides means maybe your starch or something like that. Polysaccharides carry negative charges which attract hydrogen ions moving out of cells by the action of proton pumps. These hydrogen ions are then accessible to be exchanged for soil solution cations, these hydrogen, which thus becomes electrostatically bound to the cell wall. So now your cations will come in contact with the cell wall. This hydrogen will help to bring potassium or to bring your calcium, something like this. The binding sites lie not just on outer surface of walls, but inside the capillary space within the cell wall. So there they will be exchanged. There is also the possibility of some cation exchange termed contact exchange between hydrogen ion adsorbed on the root surface and the cations adsorbed onto the solid colloidal soil particles, means exchangeable ions. Anions have few adsorption sites in cell walls. They must simply diffuse into water-filled capillary spaces. So anions can enter the cell wall spaces through the water-filled capillary spaces of the cell wall before passing through the plasma lemma. Means anions can enter through diffusion. One more diffusion has come through which the anions can enter the cell wall spaces. To some extent, they follow the cations by electrostatic attraction. There may be hydroxyl ion sometimes which can exchange for anions. The adsorption of ions may then be succeeded by absorption passes through plasma membrane. Now, they must pass when they have entered the intercellular spaces in cell wall. Now they must pass the next barrier, which is plasma membrane. There is an important difference. Adsorption is attraction outside the plasma membrane. Absorption is actual entry into the cell through plasma lemma or plasma membrane to the inside of the plasma membrane. When the ions are taken up to a greater concentration inside the plasma membrane than outside, this is termed as accumulation. When ions are taken up to a greater concentration inside the plasma membrane than outside, this is termed as accumulation. Means you get higher concentration inside the plasma membrane than outside. Then you can say some extra ions have entered the cell, and that is your accumulation. So now your ions have come to the inside the cell. So just to get more clarification here, you can see uptake ions within plant body. That is your uptake. Adsorption outside plasma lemma in free spaces, apoplast. This is your adsorption. And then absorption is inside plasma lemma in simplast. How they enter the cell, how they pass the plasma lemma. And then accumulation happens, where internal concentration inside the cell is greater than external concentration. Ion absorption by the root is the broader picture of ion uptake and transport. Now you see some more broader picture of ion uptake and transport. Ion absorption by the root is more pronounced in the root hair zone than in the meristem and elongation zones. The root hairs are simply extension of special epidermal cells that greatly increase the surface area available for ion absorption. An ion that enters a root may immediately enter the simplast by crossing the plasma membrane of an epidermal cell, or it may enter the apoplast and diffuse between the epidermal cells through the cell wall. So it can have two mechanisms. One by simplast can enter the cells via plasma membrane and can just pass through the apoplast in the interspaces between two cells, between plasma lemma and your cell wall. That is your apoplast. Now you can see some Routes of water and ion transport across various regions of plant roots. You can see they enter here. They enter here. One is apoplastic, your green one. Your green one is your apoplastic, where it is just passing through the spaces between cell wall and cell membrane, and it can be symplast. Symplast means it can uh, cross the plasma membrane. It can enter the cell wall, then pass through the interspaces, then enter the cell of the root hair. Then enter the root hair are single celled. Then it enters, and from the root hair, it can pass through the cortex and through symplast. It will go through symplast, means this from cell to cell, you can see. And every time it has to break the barriers of cell wall, but cell wall also got some plasmodesmata that can allow the passage from one cell to another cell. So likewise, you can see, बहुत मेहनत करता है. It is putting so much of labor and so many hurdles and restrictions for movement of ions across the root. And you can see then one more hurdle is your this kind of layer, Casparian strip. It gives very high resistance to for movement of ions. So in the end, they reach the xylem vessel. Now from here, they can go up in the plant system. A broader picture, again, you can see from apoplast of apoplast of cortex, and ion may either be transported across the plasma membrane of a cortical cell, thus entering the symplast, or diffuse radially all the way to the endodermis via the apoplast. So it can diffuse by apoplast also. You can see the green one, this apoplast. These interspaces. And in the end, you can see the apoplast, these green ones. Apoplast forms a continuous phase from the root surface through the cortex. They make a continuous phase. You can see this. They are touching each other at one point and like this. Apoplast forms a continuous phase from the root surface through the cortex. However, in all cases, ions must enter the symplast before they can enter the stem. Means finally, initially they can travel through apoplast up to xylem or Casparian strip. But after that, those ions who are just who are walking through the apoplast or just living in apoplast must enter the symplast if they want to be part of the ion uptake, or if they want to go reach the xylem, then in the end they must pass through the symplast. However, in all cases, ions must enter the symplast before they can enter the stele. Stele is the central part where you find xylem because of the presence of Casparian strip. So what is stele? Stele is central part of root or stem containing the pericycle and vascular bundle. Containing the pericycle and vascular bundle. That is also called as stele. जो खोखला पार्ट होता है बीच में, उसके साइड in that side you have xylem and phloem. That is your stele. So here you can see this green one. See apoplastic pathway. See it critically, the red line. It is through apoplast. It is in the interspaces passing from one to another. You see, it is apoplastic movement. It is passing through the cell, between the cell wall and cell membrane, from one cell to another cell. You see this path. But in the end, here where you find number three, it has gone because it is apoplastic up to here. But from here, it must enter the cell. It must go inside the cell, and from this cell to the xylem cell. So you can see in the end, apoplastic has to be symplastic. Once an ion has entered the cell through the symplastic connections across the endodermis, It continues to diffuse from cell to cell into the xylem. The Casparian strip is a suberized layer that forms rings around cells of the specialized endodermis and effectively blocks the entry of water and solutes into the still via the apoplast. ये मजबूर कर देता है कि आपको अब सिम्प्लास्ट में ही जाना पड़ेगा. This Casparian strip. Once, perhaps the last slide for today, once ions have been taken up into the symplast of the root at the epidermis or cortex, they must be loaded into the tracheids or vessels. Whatever cells are there in xylem, they must be loaded into that of the cell to be translocated to the shoot. Then they will start upward movement or upward transport. First is cross-sectional or horizontal movement, then your vertical movement. The presence of Casparian strip allows the plant to maintain a higher ion concentration in the xylem that exists in the soil water surrounding the root. Then these Casparian strips are also useful because they will not allow, in general, the back entry of ions who have reached the xylem. So finally, you can see this picture is very interesting. You try to understand this picture. And if you need, I can send you some special notes on ion uptake, or I can give you some references if you really want to learn in detail. So this was simply to make you understand actually what happens. And once we understand this picture, this particular picture before you, then it is easy to understand different theories, active movement, passive movement, and carrier theories, that theory, a variety of things are there. Then it will become much easier for us to understand these kind of things. So now the real thing comes, your nutrient uptake and transport in plant cells. Uptake is absorption and then accumulation and then transport from your cell wall up to the xylem. Nutrient, so you have got nutrient uptake by root and transport of movement. So tomorrow, if you have 1 hour time, then tomorrow I can explain you from 11 to 12. So you can open your videos and ask questions. Yeah. Only six participants are there. No problem. I don't bother.